is going to break apart the DNA. Helicase is a turbine that spins about as fast as a jet engine. Almost 10,000 RPMs. This is happening in every single cell in your body that is dividing. That means almost all the skin cells you have on the surface of your body, right now, there's a tiny little turbine that is spinning at 10,000 RPMs.
We should put little leashes on them to harness the energy.
That is exactly how they figured out it was there. They put a little leash on it.
So they're spitting.
They put a little tiny leash on it, and they put GFP on it, and they watched it go.
This is only happening to
Wild
DNA?
speculation.
What?
This is only happening to DNA.
DNA to DNA.
Okay.
We're making DNA from DNA.
Wasn't GFP only found like 2007? Is that like this like really recently?
All of this is really recent, yes. Okay, so topoisomerase will exist somewhere downstream. It is going to be hugely important later when we talk about cancer therapies next semester. And what it's going to do is what we demonstrated in class. Remember, where's the green thing? Where's the green thing? Where's the green thing? How do you have this? Okay, so Dean, Amelia, can you hold the end? All right. So if I have helicase and it starts to pull us apart, you'll notice that these spirals on either side become tighter and tighter and tighter. And if they don't relieve the tension by untwisting it a little bit, the DNA will break.
That's like
So topoisomerase relieves that pressure.
Right. Topoisomerase relieves that pressure. In cancer therapy, I'm actually going to try to destroy cancer cells, so I'm going to disable topoisomerase.
That wasn't on the camera. Do you
That's
want?
fine.
Yeah.
Okay, it's good.
Are we? Yeah.
What happens when you break DNA?
You don't replicate.
That's bad.
Not necessarily.
Or if it's cancer, it's good, I guess.
Right.
Are there any like diseases where people don't have topoisomerase in like normal cells? They just don't.
I don't think you would survive.
Please.
Okay. So there's an RNA polymerase that lays down an RNA primer. I'm going to make RNA dotted.
Any of their colors? You want to just make RNA green?
Let's make RNA green.
Take the soccer.
Thanks, buddy. I've had some pretty good catching in here.
Oh God, Jesus.
Okay. RNA is first laid down.
Why RNA not?
Never ask why. RNA is laid down because there is an enzyme that lays RNA down first. Why is that enzyme part of this process? Probably because all of this was originally an RNA-based system that evolved to then later on handle DNA. What is the name of the enzyme that lays down the RNA primer?
primase.
No. Gavin, it's primase.
Might be.
Okay. Where in PCR did we get the primer from?
Good people at IDT.
The good people at IDT DNA. What was our primer made out of?
DNA.
Nucleotide.
No. It was made out of DNA. From the good people at IDT DNA.
No.
Okay. After this primer is laid down, DNA polymerase is able to start working. What did it need? What did it need as a point to start? It needed that 3' end.
So what enzyme is that?
DNA polymerase now.
It was just adding new nucleotides, or just chilling.
Mm-hmm.
And this makes mistakes?
Yes. All right. there will be another nuclease that goes back and destroys the RNA and replaces it with DNA. Time? How much time do I have?
15 minutes.
13 minutes left. 15.
Is that piece of RNA
It's called an RNA primer.
So if the body goes back and destroys the RNA after, why doesn't it just figure it out and use DNA primers instead?
Because God didn't tell the body to do that.
You don't answer biology questions with because God.
Well,
Wait, and then what does life
One of
That's
us was given a
magic.
$250,000 grant.
Okay. So
Wow.
teach with the power of love, Luke. not
Hit him with
And
it.
God.
What?
No. Okay, so why, the question is a very good question. Why does this not make sense? You don't think this makes sense because you can't see the previous version.
Yeah.
If the previous version was all RNA,
Right.
So it's just like unfinished.
then maybe this DNA polymerase was an RNA polymerase to begin with, and it adapted to pick up DNA.
Ooh.
So maybe this is the advanced version, not the backward version.
How does a primase that adds the RNA prime end thing? How does it know how much to add or when to stop?
That's okay. So that's a really good question. So another thing you have to realize. I'll show you. You're not gonna like it. But I'll show you. Yeah.
Where does
So you
RNA?
said that, sorry.
No, you go. I don't know.
So you said that something like goes back and gets rid of the RNA primer and replaces it with DNA. What's the name of the protein that does this?
It's an exonuclease. Removes the RNA. And the thing that it replaces with is a DNA polymer. So what enzyme would put it? What
DNA
makes?
polymer.
DNA.
Yeah, it's another
polymerase.
DNA polymerase. Again, all of this information is in that article.
So
Yes,
what you're explaining is how you get from DNA to RNA.
DNA to DNA.
Right. Yes. This is replication.
This is replication.
Okay.
So there was RNA involved, but we end up with DNA.
Would you mind drawing the RNA?
I did. It is
Yeah,
now
you
replaced by the
Oh.
exonuclease.
Oh, wait. How? Yeah, that's the thing.
With
Okay,
another-
so
Sorry, I must have been writing something.
all right, let me just do it again from the beginning.
Sorry.
Okay.
From
So here we go.
Sorry, I was like.
The helicase actually has a couple arms. It's going to be attached to two different proteins. Okay, one is going to lay down an RNA primer, which is going to allow DNA polymerase to attach to it.
That all makes sense.
The RNA is removed by the exonuclease, and DNA. Oh, that's wrong color.
So exonuclease is a type of DNA polymerase.
No.
What?
A separate DNA polymerase comes in and adds its back. Why? This process probably evolved from the production of RNA.
So is RNA still reproduced? Or?
Kind of.
Is it important?
Later. Okay.
Where did the RNA come from that was put in?
It's
floating around?
in the solution.
chilling.
Yeah, you know what? I actually did a lot of research into that because I've never been satisfied with that answer, but it does just look like it's. It literally looks like it's floating around.
Okay, good to hear.
Okay. Now,
I'm sorry, what was the thing called that kills the RNA and replaces it with DNA?
exonuclease kills, DNA polymerase builds. Okay. All right. So now, this is going to be a little hard to conceptualize.
Okay.
We can only build DNA in one direction. DNA polymerases only add to the 3' end. So it will be no problem on one strand to add DNA. The polymerase will travel that way.
What about the other strand?
Yeah. So that's a problem. What's that? So let's look at what's going to happen here in 1 second. The helicase, actually we're going to feed the DNA through. Okay? So what I'm about to draw is going to be the DNA fed through. So this, I'm going to move over here. Everybody see that? Okay. We're going to push the DNA through. This is going to move over here. I'm going to push the DNA through. This part, this copied part, is now going to be right about there. Okay? Watch. Pushing DNA through. DNA got longer. Everybody see it? Yes?
This is really good. For some reason, I really stuck on the idea of the last thing. And
Can we come back to it?
but it's real. It's helpful.
Okay. I promise it's on video. You can.
Oh.
Right? So, we're going to do the same thing again. This is why I was saying, just hang on a second. We're going to do the same thing again. An enzyme is going to lay down a primer. What's that enzyme called?
Oh, primase.
Primase. That's going to allow an enzyme to make polymers of DNA.
DNA polymerase.
But it's DNA.
And then there's one that joins those two together, right?
Well, first we have to get rid of.
That answers my question.
We have to get rid of the RNA,
With exonuclease.
exonuclease.
And then polymerase fills it back in with DNA.
Polymerase fills it back in with DNA.
And then it gets pushed through.
Actually, there's going to be one more little step.
Joining those two.
Ligates. You mean?
Oh, like.
Do you see this gap?
Ligates.
We're going to need to ligate that. Glue it together.
So that's why. That's how.
What about the
That's
other
how
end
the
of the
space
DNA?
with the RNA primers then gets filled in because it needs a 3' end to build off of. So there's another RNA primer put further down on the strand and then it gets filled in and DNA gets stitched in. So that's why you it replicates and it goes back.
So basically what I'm doing is imagine that I'm copying letters on the floor in front of me. I'm copying a little bit. RNA gets removed. RNA gets replaced with DNA. Then I start back here. Read to where I was. RNA gets removed. RNA gets replaced with DNA.
Kind of like backwards leapfrog.
Yeah, kind of like backwards leapfrog. Yes.
What happens when it keeps going back and it copies the whole line or segment until it reaches the next origin site?
So it's going to copy until we reach the next origin, yes.
Okay.
And then ligase will join those two segments together.
Okay.
All right. The other side is actually a lot easier. It's going to be this complicated process. Let me explain it to you.
Oh god.
Ready?
Wait.
Okay, just wait. First, we're going to add an RNA primer.
Okay, the stuff, Luke.
Then, an enzyme is going to add DNA to that RNA primer. Essentially, it's going to polymerize DNA. Its name is
DNA polymerase.
DNA polymerase. Okay. Then, I know this is confusing and very different, but the RNA is going to be removed by, does anybody know?
Exonuclease.
No, an exonuclease.
Yes.
Luke, you're killing us, man.
Okay, here's the only difference, and this is what makes it actually easier.
Can it just keep going?
Right. So I'm going to force more DNA through. That means this piece is going to move to down here. Everybody see that?
Yeah.
This piece is about to move here. This piece right here is going to move here after I take this DNA and move it here.
So you're feeding the DNA through the heel.
Feeding the DNA through the helicase.
So you don't need any ligase?
Not yet.
Is that kind of like vegetable peelers that you like force the vegetable
Yes,
through
actually I
and
can
it
kind of see it.
splits open?
Very much smaller.
Yeah.
They don't create new
That didn't make
vegetables.
sense to me until the vegetable.
Okay, everybody see this? Hey, don't lose me yet. Don't lose me yet.
We got you.
This is like the last part and we're done.
That's good to hear.
Okay. Do you see here that our enzyme is building this way?
Yes.
Do you see that we have a 3' end?
Yes.
So the enzyme just continues to build.
Until it reaches the next origin site?
Yes. Until I feed more DNA through. And then it just builds more. Does everybody see that?
Yeah.
So this one leapfrogs. This one does not leapfrog.
Pretty wild.
No idea.
There's no ligase, no.
Good hear.
Yes.
Is the only time that it can make mistakes is when it's the DNA polymerase is building the...
So when the mistake will happen will be when the DNA polymerase is lining up.
Only then, like that's
Yes,
the only
because
time. Okay.
you've got it.
Is that it?
Yeah, I do. I've got 3 minutes left in this class.
Okay. No. Are these the big important ideas?
No.
No. What are the 3 big important ideas?
It occurs at origin.
Toby.
Let me correct my page. Okay, so first the replication has to happen at an origin point.
Right.
What we saw with like how you have to.
What is the origin point? Can you tell me?
So origin point is a sequence of DNA in the DNA strand that like tells the whatever it's called the representative like where to start.
Okay.
replicating the DNA.
What's the metaphor for the origin?
It's like when you're reading a book. Like if you're reading, sorry, if you're trying to completely copy an entire book, right?
Yes.
You will like start at a chapter and you'll copy the chapter and then you'll leap back to copy another chapter.
Right. The word chapter.
The word chapter, yeah. Start at the word chapter.
Right.
And then like you copy that entire chapter, starting at that word chapter. When you go back to the next instance of chapter, you copy it from there. Go back to the next instance, so you copy that chapter.
Okay. Cool. What is the second most important idea? Yes. Give me 1 second. Let's finish this.
The origin complex of replication, which is all of this, copies DNA at the origin.
Okay.
You kind of said with the top 2 points.
Yeah. All right.
The third point.
Okay. Who hasn't gone yet? Tom.
It makes mistakes, which can be bad, no change, or better.
It makes mistakes, which would be bad, no change or better. Which one is most likely?
No
No
change.
change.
Which one is then most likely?
Bad.
Then?
Better.
Right? So
But it's good to have
the messed up part, here's super irony, and I'm gonna leave you with this, right? So
questions.
okay, alright, I won't leave you with this. The DNA polymerase was likely at one point an RNA polymerase. And the gene for the DNA polymerase, the gene for the RNA polymerase, got screwed up so that the RNA polymerase started picking up DNA.
Humans exist because of mutation.
So it messed up and got better.
Messed up and got better.
Yeah. Is that evolution?
We don't know what that word means.
We don't.
That is when genes
Is that
mess
an
up and
example?
get butter. So the reason I can't explain that to you yet, Chris, is because evolution does not happen to an organism or to a molecule.
Happens to.
It's a step toward evolution.
No, evolution happens on a much larger scale.
It's a mutation.
A mutation is what we are discussing.
Yeah. Okay. Cool.
We are discussing a mutation, which is definitely a part of evolution. Okay, Bean.
Okay. What Toby was saying about the chapter book, does that not happen on this
on this slower side.
Yes, but we're just showing one chapter. So there's actually another replisome over here doing the same thing.
Okay, so you
Different
said no
origins
ligates in that part. So it's
And
not
the
a
bottom
book,
strand.
It's just a book with no chapters. It's just like a block of text.
No. So if I was copying the book,
Yes.
there's two ways of copying the book. I can copy from the bottom and kind of go like this.
Or you can just
just
read
go
it top
straight
to
through.
bottom.
Or straight. And this is a little bit where the metaphor falls apart because we would need double-sided pages somehow.
Going the opposite way.
Right. It like the cookbook will work for us, but
Can you put it in backwards?
yeah.
Dude.
I can read backwards sometimes.
All right.
Just
Fundamental idea in biology. DNA is replicated. This occurs during basically 2 times: when you're growing or when you're repairing, anytime you're making new cells. That is part of a cell cycle. It is not mitosis, which is a common misconception. There are many enzymes that work here. A series of enzymes containing helicase, or ones that attract helicase, will bind to the replication complex, or to the origin of replication, which is a sequence of DNA that's called the origin of replication complex. They will actually attract helicase. Helicase will pull apart the strands of DNA. An RNA primer will be laid down by an enzyme called RNA polymerase. DNA polymerase will then pick up from there and build DNA. The RNA will be removed by an exonuclease, replaced by a DNA polymerase. In one strand, that's going to happen multiple times, and those pieces are going to need to be glued together by an enzyme called ligase. Everything we've learned about dehydration synthesis applies here. Everything we've learned about enzymes and active sites applies here. It's every single thing I have taught you up to this point is connected. What was our lab? In our lab, we did 2 types of replication. We classify them by where they were done.
In vitro.
In vitro and in vivo. What was the in vitro version called?
PCR genesis.
Right. PCR mutagenesis, and in vivo, which we just call it replication. Jerry.
Or
Yeah.
bro, Jerry.
It's like our lab is this class.
Wow.
Hey.
Am I the only one that gets excited? I'm really excited.
I'm too tired
You know,
to be
it
excited.
was
at
like
this
a
point.
lot of planning, and I was just sat there for about 2 weeks like, this is not going to work. This is
It's
not gonna work.
3:32.
You get out of here. Go ahead.
You guys-
Luke, you just
Luke,
talked to our
next.
soccer practice.
Is the video over?
What?